Pollination is an essential process in the production of fruits, vegetables, and forage, as well as in the production of seed for many roots and figure crops. Some plants and flowers can self-pollinate, but in many others, cross-pollination occurs in which pollen is transported from one flower to another, either by wind or animal action. For example, zoophilic pollination, the latter being the most frequent and efficient case. It is estimated that within the 90% of pollinations that occurs in flowering plants worldwide, 67% is carried out by insects, constituting the most important group of pollinators, both for wild and cultivated plant species, which means that they are fundamental in the maintenance of ecosystem and global biodiversity.
In Chile, as in most of the world, pollinating insects such as bees play an invaluable role in agricultural production, as they not only pollinate flowers, trees, and other types of plants, but also provide food and habitat for other species, which has a direct impact on the country's economy, biodiversity, and food security. In recent years, native pollinators' populations have declined significantly around the world. In Chile, bees are threatened mainly by human activity through the inappropriate use of pesticides in crop fields. Although pesticides are widely used in agriculture to protect crops from disease, weeds, or insects, simultaneously ensuring an increase in agricultural production. Compounds such as neonicotinoids present in some agricultural pesticides can have disastrous consequences due to their highly toxic and lethal effects on bees. Neonicotinoids, being structurally similar to nicotine, have a similar effect as agonists of nicotine acetylcholine receptors, thus directly affecting the bee's nervous system, producing memory problems, abnormal behavior, decreased mating frequency, and immune system failure. Because of this, pesticide use has been considered to be among the top 3 factors causing the decline of pollinators such as bees almost worldwide, with neonicotinoid pesticides being a group of contaminants of particular concern.
Based on this, we propose a team from Universidad Andrés Bello to use synthetic biology to create an heterologous biological system, which can sense the presence of neonicotinoids and, in response to that, start the separation of this volatile compound called geraniol, which can be sensed by the bee and makes the bee temporarily move away from the crops when this pesticide is active. This volatile compound is geraniol, a monoterpenoid that makes up the majority of rose and citronella essential oils. The base structure, like the other terpenes and isopentanes or isoprenes, such as isopentenyl pyrophosphate and dimethylallyl pyrophosphate, 5 carbon-branched compounds that can be synthesized from 2 synthetic roots. On the one hand, the mevalonic acid root, and on the other, the methyl erythritol phosphate root respectively. Therefore, we need a biological system containing an endogenous isoprenoid pathway that allows the heterologous production of geraniol. In this sense, we will use a chassis, a transposon 5 mutant of Azospirillum brasiliense sp7, a non-photosynthetic strain, but which is a superproducer of carotenoids in the presence of light, CAR1, and whose genomic analysis of this bacteria strain for the isoprenoid pathway revealed that it possessed all the genes involved in the production of isopentenyl pyrophosphate and its isomer, dimethylallyl pyrophosphate, where from isoprene it is possible to form geraniol diphosphate, and from these polyterpenes such as carotenoids, a synthetic root in common that can be exploited for the high synthesis of geraniol. But not only that, being a commercially active species and having a close interaction with plant roots, it can establish a mutually beneficial symbiotic relationship without bringing negative effects to the plant. Therefore, Azospirillum brasiliense, which is already found naturally in the treatment of crops, would be an ideal chassis for the heterologous production of geraniol.
The chosen chassis is the bacterium Azospirillum brasiliense, since this is a rhizobacterium capable of promoting plant growth of different crops of agronomic interest, in addition to belonging to the macrobiota present in the roots of cherry crops. To carry out this proposal, we will use a mutant of Azospirillum brasiliense sp7, which is a superproducer of carotenoids in the presence of light known as CAR1, and which has a synthetic route that can be used for the synthesis of geraniol. However, only two enzymes are required for this: geraniol diphosphate synthase and geraniol synthase. Therefore, in order to induce geraniol synthesis in the presence of neonicotinoids, a genetic circuit is required. The constitutive expression of geraniol diphosphate synthase, the constitutive expression of the neonicotinoid receptor capable of activating geraniol synthesis, for which we will use an odor binding protein 2, OBP2, which upon binding to its ligand, in this case neonicotinoids, undergoes a conformational change which could be used to modify its structure so that when they bind, this conformational change exposes a DNA binding region, DBD, capable of acting as a transcription factor, for which we will use a zinc finger DVD, and the expression of geraniol synthase inducible by the modified OBP2. Among the parts that we will need, some already exist, which are a strong constitutive promoter, a strong RBS, a strong terminator, the prefixes and suffixes to standardize the parts, as well as the two missing enzymes for geraniol synthesis. And the parts we will have to create would be the modified OBP2 and the inducible promoter inducible by the modified OBP2 in the presence of neonicotinoids. To be able to create them, we searched the NCBI database for the sequences of an OBP2 and a zinc finger DNA binding domain, from which we obtained their amino acid sequences and then obtained the nucleotide sequences. Then we encountered a problem. When analyzing the parts of the parts registry for the enzymes geraniol diphosphate synthase and the geraniol synthase, we found that they had several stop codons in their sequences, so we could not use them to synthesize these enzymes. Therefore, we searched the NCBI database for sequences for these enzymes and found amino acid sequences that we could use. Having the necessary sequences, it is necessary to optimize these sequences for our chassis Azospirillum brasiliense. For this, as this organism was not found in optimization websites, using the frequency of each codon found for this bacterium, we were able to obtain the optimal DNA sequence for this protein based on their amino acids. Then, having the sequences of the necessary parts, we organized them by means of the benchling tool inside a vector, in this case PSB1C3, which contains the chloramphenicol resistance gene. Within this, in its multiple cloning zone, we introduced the three parts necessary for the circuit, with the prefixes and suffixes RFC10, and making sure that the restriction sites present in these sequences are not found within the parts. Finally, we simulated the circuit in the Tinkercell tool. For this, we divided the circuit in two parts. First, we tested the part corresponding to the expression of the enzyme geraniol diphosphate synthase, obtaining a good expression of this in time in the deterministic simulation, as in the stochastic one. We then tested the part corresponding to the expression of the modified OBP2, its binding to the neonicotinoid, as well as the induction of geraniol synthase expression. We can see that in the circuit, the expression of geraniol synthase depends on the promoter activator, which in turn depends on the transcription factor resulting from the binding of OBP and neonicotinoids. By simulating this, we obtain an acceptable rate of expression of the modified OBP2 and geraniol synthase, the final enzyme in the circuit, and we were able to see a reasonable expression of this in both the deterministic simulation and stochastic simulations.
Considering that bees are the main pollination agent and that they also have an effect on what will be biodiversity and ecosystem formation, who this will go hand in hand with the 2030 agenda for Chile compared to the other countries in the world where these neonicotinoid-based pesticides are not longer used.
Ya, a Chile y al mundo le interesa cumplir con los objetivos de desarrollo sostenible, pero compuestos. Sin embargo, tenemos que ser súper conscientes de que el uso de productos fitosanitarios como los neonicotinoides tienen un papel importante en la producción de fruta, que es limitar y controlar las plagas que producen un daño económico también grande en la agricultura. Sin embargo, es necesario supervisar de mejor forma su aplicación y su uso. Y quizás sea importante generar nuevos datos como los que ustedes pudieran obtener a partir de su proyecto, nuevos datos que den cuenta de los efectos nocivos de un mal uso de estos productos, de los neonicotinoides, y así avanzar en el cumplimiento del principal objetivo de desarrollo sostenible del cuidado ambiental. Por un lado estamos limitando la mortalidad de las abejas, por otro lado estamos limitando la contaminación de las aguas, estamos limitando además la contaminación de los suelos. Entonces no solamente tiene que estar en nuestro enfoque las abejas, sino también todo el ambiente agrícola y el paisaje agrícola en su conjunto. Porque por un lado están los agricultores tratando de exportar más fruta, que Chile es un gran exportador, productor y exportador, pero además por otro lado están utilizando para promover aún más la producción y la exportación de fruta, utilizando muchos compuestos que pudieran ser nocivos para las abejas y para el ambiente. Entonces, ¿cómo armonizamos? Y que siempre el desafío que también nosotros nos planteamos en nuestro trabajo, ¿cómo armonizamos producción con conservación del ambiente? Entonces, yo creo que en ese sentido este proyecto pudiera dar luces de cómo mejorar el ambiente para las abejas, mejorar el hábitat para el buen funcionamiento de las abejas nativas y melíferas, y por otro lado no dejar de producir fruta, que es un ingreso económico importante para Chile.
Therefore, based on the 2030 Agenda, the SDGs goals for this project corresponds to goals 9, 13, and 15. The Sustainable Development Goal 9 build resilient infrastructure, promote inclusive and sustainable industrialization, and foster innovation, aligned seamlessly with the proposed project, as it advocates for sustainable industrialization and investments in scientific research, both of which are significant measures to facilitate sustainable development. The Goal 13, Climate Action, aims to combat climate change and its effects. Therefore, by promoting the healthcare of pollinating agents such as the Apis mellifera bee, the increase in pollination levels will help increase the formation of ecosystems and thus decrease the high levels of carbon dioxide. And last but not least, the Objective 15, Life on Land, seeks to stop the deterioration of biodiversity by human action. Therefore, by avoiding the toxic and lethal damage suffered by bees due to inadequate use of neonicotinoid-based pesticides, we are protecting one of the key links in biodiversity and the maintenance of ecosystems.
Our project does not have a negative footprint on the environment and offers an attractive solution to protect pollinators like bees from the toxic effect of neonicotinoids-based pesticides. The same solution can be applied to produce other biocontrol molecules in the field, leading to a broad application portfolio of solutions for the agriculture.
The projects must follow the guideline of the Bioethics Committee of your university. In addition to following the norms described in the SEM Solution 892 of the Ministry of Health and approved by the Agriculture and Livestock Service.
It is ideal to find a balance between the floral resources that exist in the field and the amount of pollinators that are acting in order to have a sustainable production.